everyone, my name is Shahana Williams, and today I'll be discussing my TAB proposal topic titled The Modification of DNA and How It Contributes to the Criminal Justice System. What is DNA? DNA is an acronym known for the term deoxyribonucleic acid. Deoxyribonucleic acid is a self-replicating material that is present in all living organisms. It is the main constituent of chromosomes that exist in every human being. Each individual on Earth have a unique form of DNA, with the exception of identical twins. This makes it easier for the criminal justice system, in addition to prosecutors, to be able to actually place a person at a crime scene if they left a certain form of their genetic information at the crime scene. In other words, DNA is the carrier of genetic information. DNA technology. DNA technology was first discovered and used in England in 1985. DNA is an important tool that is used in criminal cases. The first case to ever use DNA technology and actually convict a subject using DNA was used in the United Kingdom in 1988. DNA is the most important form of evidence within the criminal justice system. The system relies on DNA samples in order to assist in solving criminal cases. DNA testing is mostly used in criminal and civil cases. DNA has evolved since its first development in the early 1980s. When DNA testing first developed, it took forensic scientists 2 to 4 weeks to get the samples tested and back from the lab. This time constraint actually was a delay because the offender or suspect could get away before being proven guilty of actually committing the crime and being present at the crime scene. Now that technology has advanced in this process, it takes no more than 2 days in order to retrieve samples back from the lab that are considered reliable and efficient information. What are DNA samples? There is a variety of DNA samples that can be collected from a crime scene. These samples range from dry blood stains, dry saliva stains, sperm mixed with vaginal epithelial cells. In this case, the sperm mixed with vaginal epithelial cells are in cases such as rape. There are also other samples that can be collected from a crime scene, such as hair samples. Hair samples can be used as DNA because of the follicles that attach the root of the hair to the scalp, which in turn, if it is physically pulled out aggressively, it may have tissue from the root that's attached. This root will contain blood on it, and the blood is a sample of your DNA. When hair is forcibly pulled out, it can be interpreted by the forensic scientists as some kind of physical violent altercation that occurred at the scene. When hair falls out naturally, it has a club shape. However, if it is forcibly pulled out, the root of the hair will be broken or stretched, and it may have tissue, as I stated before, from the root that is attached. DNA relation to the criminal justice system. The collection of these samples is very important. They provide central evidence when dealing with criminal cases that involve serious crimes such as murder, rape, theft, and even assault. DNA evidence provides answers to the following questions. What happened at the crime scene? How did the murder, rape, or theft happen? When did the crime scene happen? Who was involved in the crime? Which is the most important question. These 4 questions are essential when solving a case. Prosecutor's favorite. The prosecutor's team consists of police officers and forensic scientists. Prosecutors and police officers have a strong relationship with one another. Prosecutors depend on police officers to help them close their case and convict the suspect. DNA analysis is one of the most important factors used in the criminal justice system. Without the use of DNA samples, prosecutors may have to use other forms of evidence, such as videotapes, witness identification, and interviews of people that know the suspect or victim. These forms may create bias. Information such as these are not reliable or as reliable as DNA and can lead to mistrials, wrongful convictions, and even criminal cases that are dismissed due to lack of evidence. DNA is such an essential tool within the criminal justice system that prosecutors uses at least 75%. 75% of their cases are based and solved based on DNA samples that have been collected from a crime scene. Out-of-body DNA. This relates to the relationship between police officers and the prosecutors and the forensic scientists. Police officers often play a big role when trying to obtain DNA samples from a suspect who is being investigated. These ways of collecting DNA often occurs when there is no DNA left at a scene. This is known as DNA biologics. DNA biologics are human cells. Human cells carry a nucleus. This nucleus can survive departure from the body by itself. DNA can travel from a suspect to victim, from a victim to a suspect, and most importantly, from a suspect or victim to an object or location. Due to DNA in its nucleus being able to travel from place to object or from suspect to object, police officers have found a way to identify a suspect even if the suspect has not left any DNA at the crime scene. This form of collection of DNA is known as out-of-body DNA. Police officers are more reliant than ever on obtaining out-of-body DNA, and so are prosecutors. This out-of-body DNA and evidence is what brings the connection of police officers and prosecutors together. The collection of out-of-body DNA. It's simple. When police has a person of interest, they invite them for an interview at the police station. They may offer them a cigarette, something to drink out of a bottle or a straw. The person of interest is then unaware that once they smoke the cigarette or drink out the bottle, that they have left a sample of their DNA on that object. When the person leaves the cup or cigarette butt in the station, the police are then able to send the item to the lab for an analyst. The results determine the outcome of the case. If the DNA sample is compared to any of the other samples found at the crime scene, the police officers have succeeded in assisting the prosecutors in solving their case. You're under arrest. Say I. The process of arresting a suspect has changed due to the modifications of DNA. When suspects are arrested, they are put through the following process. Originally, they were put through the suspects would read their rights. A photograph, also known as a mug shot, was taken, and the suspect's fingerprints were taken. Now that the modification of DNA has advanced, there's a new process called DNA extraction. DNA is either taken by drawing blood from the suspect's index finger or by swabbing the inside of the suspect's mouth, known as the buccal swab. State versus legislation. There are different legislations and statutes from state to state when it comes to extracting DNA upon the person's arrest. For instance, out of the 52 states, 23 states do not allow DNA to be taken at the time of arrest. These 23 states only allow DNA to be taken once a suspect is convicted of a crime. In New York State, it is illegal to extract DNA from a person that is merely under arrest. The person must be charged and convicted with a felony before they can undergo any DNA extraction. So if the person is convicted of a misdemeanor or a violation, it is illegal to extract DNA from them. In Oklahoma, it's the same thing. They cannot get DNA extracted from them for just a mere arrest. However, there are exceptions. If the person under arrest is an illegal alien, only then is it lawful to extract DNA from that person. When is DNA used? The first reason DNA is used is to solve a crime when a suspect is compared to the evidence that was collected at a scene. If the sample matches the perpetrator, then the suspect is likely to be convicted of the crime. Second reason, DNA is also used in criminal cases in order to solve a crime when a suspect is not identified in a case. However, if any form of DNA was left at the scene by the unidentified suspect, that sample is analyzed and compared to the offender's profile if he has been previously arrested and charged with a felony, through a DNA database known as the Combined Index System. A federal database. When a DNA sample is extracted, the suspect's DNA profile is uploaded to the state and federal DNA database. This database is known as the CODIS, also CODIS. This database was originated by the federal government in the 1980s in order for federal, local, and state agencies to use, store, and exchange DNA profiles. Everyone that is convicted of a felony is mandated to give a sample of their DNA to law enforcement. If the offender refuses to provide a sample, they are charged with a misdemeanor in addition to their original charge. So not only do they get a felony, they will also get a lesser charge, such as a misdemeanor. The quota system. As you see in the slide, this map shows the United States of America, which are the 52 states, and the key is the code in colors of how many individuals in terms of decimals are signed into the quota system. So you see the green areas have less than 75% that are signed up in the CODIS database. The purpose of the CODIS database system is to assist law enforcement with gaining information and access to offenders' records in order to compare the offender's DNA with crime scene evidence to a database of DNA profiles. These comparisons of DNA profiles help eliminate any other suspects that are thought to be involved in the case, in addition to matching the DNA to the correct offender. The CODIS system is so essential to the criminal justice system because it compares prior offenders' DNA all over the United States to DNA samples that are being tested at that specific time. DNA contributes to exonerations. Exonerations is when people are free from being wrongfully convicted. DNA testing is also used for getting people out of prison for being wrongfully convicted. Due to the modification of DNA technology, cases in which people are wrongfully convicted are reopened and investigated, proving that the person was not involved in the crime, in addition to proving that the individual that served the time did not do the crime or was at the crime scene. These cases are known as post-conviction. Examples of exoneration cases. According to the www.innocenceproject.org website, the following people were exonerated due to DNA technology. These two cases are just two out of many cases of people that have been exonerated. The first one is Kirk Bloodsworth. He served 8 years in a Maryland prison for a murder he didn't commit until 1993 when he was exonerated. The second one is Rolando Cruz and his co-defendant Alejandro Hernandez. They served more than 10 years on death row in an Illinois prison until DNA testing proved that they were innocent in 1995. So you see, individuals have also been sentenced to severe time and on death row and risk of losing their life for crimes that they did not even commit. In conclusion, DNA is not only a tool that is used to convict suspects, it has also been used by prosecutors and defense lawyers to exonerate people that have been wrongfully convicted. The revolution in technology along the years have evolved dramatically, and it has contributed to the criminal justice system in many ways. I believe that they will continue to use the system because not only is it efficient, it is also effective in getting the right person for the right crime. Thank you. If this was a live presentation, I would ask any questions. I hope that you enjoyed this presentation as much as I enjoyed researching and gathering information for this topic of how DNA technology and the modifications of DNA contributes to the criminal justice system. Thank you all.